7 o'clock. Let's get started. Thanks everyone for joining. Looks like there's a few people still joining in. We will kind of get things underway. Thanks for taking up your Wednesday night to learn more and hear more about chronic wasting disease. Cait is a frequent flyer on our conservation webinar series. We have her on once or twice per year. Chronic wasting disease is a pretty probably the biggest threat that we're facing in BC, and it's ever evolving as it does here and in other jurisdictions. I know there's a lot of interest from our membership and from the broader public, and so the goal here is to educate you, answer some questions. If you do have questions, you can put them in the Q&A, I believe, and then we'll probably get way more questions than I have time for, so we'll aggregate the questions kind of thematically and then get some responses from Cait. And if we don't have the answer, we will try to track it down if possible. Keep in mind that Cait is a biologist, not an elected official, and so she doesn't get to decide a lot of the things that happen at the end of the day. She does her best to try to do the best with what she's given, but we don't always get all of the funding and the support that we want, and that's not her job. I guess that's our job as advocates and our job to work with elected officials. So thanks, Cait. I'll do a quick intro on you. Cait is a wildlife health biologist with the Ministry of Water, Lands, and Resource Stewardship, affectionately known as Walrus. She joined the BC Wildlife Health Program in 2006, now leads priority disease monitoring programs, including chronic wasting disease and bovine tuberculosis. She's dedicated to working with researchers, agencies, stakeholders, First Nations, and the general public to ensure that these groups have access to current and accurate facts on priority wildlife issues in BC. Cait, I'm guessing your presentation is probably 40 minutes, something like that?
Yep. We'll shoot for that.
Around there. I probably added a few slides just based on the questions and the correspondence we were getting as of yesterday. So, folks, if you have questions, put them in the chat on the way. We'll ask them at the end. And Cait, thank you again for coming on a Wednesday night. We really appreciate it. We appreciate all the work you do.
Thank you so much. I really appreciate the invitation to come and share this work and the information as always. So appreciate that. Thanks for having me. Let's dive in. So I'm just going to start with some basics to lay some groundwork on why we are concerned about this disease and some facts, that to kind of guide how we approach it and why we approach it the way we do. So chronic wasting disease, or CWD, is a prion disease that affects the species in the deer family, and that includes deer, elk, moose, and caribou. The prion that causes the disease is an abnormal misfolded protein. These proteins don't function like normal proteins. Instead, they accumulate in the tissue of the animal, concentrating in the brain and causing damage to the brain tissue, so leading to a neurological disease. The image in the background here, the kind of purple colored image, is infected brain tissue under a microscope, and you can see the sort of lighter colored circles in that tissue, and those are literally holes in the brain tissue that result from this disease. So it's always fatal, and unfortunately there's currently no treatment or vaccine. Now, one of the most challenging things about this disease is its potential to contaminate the environment. Infected animals will spread disease by shedding these prions through their bodily fluids or carcasses into the environment. And other animals can then be exposed either by direct contact with an infected animal or by picking up the prions from the environment. Those prions can be in the soil, in water, or on the surface of plants. And once the prions get into the environment, they will persist for several years. They are resistant to burning, to freezing, to disinfectants. And so this creates an ongoing source of disease in the environment, which just perpetuates this cycle. From a food safety perspective, CWD is not like some other diseases caused by parasites or bacteria maybe that are often can be safe to eat after you cook at the target temperatures. That's not the case with prion diseases. Cooking does not eliminate the prion. The other really challenging thing about this disease is that most infected animals appear healthy. So we call these animals silent carriers of the disease because we rarely see animals with symptoms in the wild, and there's no signal to us that there's something wrong. So the reason for this is that it takes several months for any symptoms to start to show. Those symptoms only really become apparent at the very late stages of the disease before the animal dies. Um, so most animals that do test positive for CWD are actually healthy-looking hunter-harvested animals. Um, even if you've harvested an animal, even when you're processing that animal, there's no way to tell if that animal is infected just by looking at it. The only way to know for sure is through the testing. Now we've seen the conservation impact play out in other places where deer populations have declined dramatically, unfortunately, in some places in the States and in the prairies in Canada. Um, the disease will reduce survival rates and therefore population growth rates, but it takes some time for the disease to establish and take hold of these populations. So you don't tend to see that kind of population level impact right away. What we do see right away, and we have already experienced this in BC, is the immediate impact that this disease has on people, impacting hunting culture, cultural practices, food security, things like that. And so really the key is to prevent this disease from spreading and establishing in new areas. So we can limit these negative impacts, both immediate and long term, as much as possible. We get this question a lot, but I'm not a human health expert. I work really closely with colleagues in public health, and they are who we look to provide the guidance around human health concerns. But what they are saying is that there is no direct evidence that CWD can infect people, but there's still a lot we don't know about this disease. So public health can't rule it out completely, the potential for it to transmit to people. So as a precaution, it is recommended that infected animals that are infected with CWD are not eaten. Now, public health has provided some guidance and prepared guidance for hunters and for butchers in ways that you can reduce risk of exposure um, to yourselves. Things like having animals tested so that you can have the peace of mind that the animal you've harvested is healthy. Cleaning surfaces and tools with bleach. And once you've processed your animal, to store those animals separately so that if one does come back positive for CWD, then you can separate that easily and not risk wasting other meat. So understanding the risks around CWD and working towards mitigating those risks is really our focus. There are three main areas of risk that I'm going to outline here. Disease spread can occur through live infected animals moving on the landscape, but it can also move by people transporting infected carcasses or contaminated material like plants or hay. And it's that human movement that's really most concerning because that material can travel much longer distances than, natural animal ranges. And so the disease could pop up in areas that we don't expect. The education around this is super important so that people understand those risks and the role that they play, that the people play with their behavior around these risks. If CWD is introduced to an area or a population, we need to limit disease transmission as much as possible. This involves, or the best way to do this is to remove infected animals from the system, to remove those infected animals from the landscape, mainly through hunting is the primary tool that's used for this. The other thing that we want to think about with disease transmission is limiting the potential for CWD hotspots. that are created by, aggregating animals around attractants or food sources. Urban deer, for example, are really high risk because they tend to have very high rates of disease transmission just because of their behavior and their ecology. And the third risk highlighted here is human around human exposure, that human health risk. But I'm going to defer to the public health folks to address those concerns. And if anyone has any questions, I can point you to those resources. So the good news is that we're not alone in this fight. We've learned a lot from the experience of other places. CWD has been affecting places across North America for quite a long time. So here are some of the key learnings that we've from the knowledge that has been generated from some of these experiences in other places. Early detection, detecting that disease early when it first is introduced to an area is critical before that disease is allowed to establish and get into the environment. Precision actions, so really focused actions that are based on sound information are more effective. Knowing where and when to focus efforts are much more effective and generally much more accepted. Sustained CWD management is really important for the long term to have any meaningful effect. And key to all of this is meaningful consultation with hunters and communities so that we can build trust and maintain that support from these communities, from hunters in particular, because hunting is really the most important tool that we have in CWD management. We need to do this together. We need to work together on this. So these learnings are really the principles that guide our program here in BC. Now we all know of examples of what happens when management did not work. This has been well documented in places like Alberta and Saskatchewan and Wisconsin, with increasing disease prevalence, continued spread, and declining populations. The disease is now well established in these places, and there's little that management can do to try and contain or control it at this point. We do continue to learn from their research and their monitoring, but we here in BC, we do not want to end up in the position that they are in today. I think we can do better in BC. And thankfully, there's also some good news stories that we can learn from. Illinois is an example of where management has worked. So how did they do it? Well, right when CWD was first detected in Illinois in the early 2000s, the Illinois wildlife managers sat down with hunters to develop a plan together. They applied this two-step approach with hunter-led surveillance, first to identify the cases, and then they followed that up with a combination of special hunts and some agency sharpshooting in specific areas to remove the animals that were at highest risk for being infected. And by these methods, they were able to remove 4 times as many infected animals relative to just sort of the general hunting season. So really effective at slowing down the disease and preserving hunting opportunities in these areas over several years. Now, Illinois is very different from BC. It's a very different system. They have very high density white-tailed deer populations, and around 150,000 whitetails are harvested in the state every year. So it's a different scale. It's a different system from BC, but I still think that there's we can learn from their approach. They are starting to observe a bit of an uptick in their disease prevalence. You'll see on that graph, it's just starting to creep up a bit. So they're reevaluating their management and kind of refocusing in certain key areas to address this. But even still, they're sitting at around a 7% prevalence. And although that's much higher than we would ever want to be in BC, we're hoping to keep things under 1%. This is still a much better outcome after, 20 years than some of the prevalence rates that we've seen in other places, like 10, 20, 30, 40, 50% prevalence that we've seen in other places over the same time period. So Illinois is doing something right, and I think we can learn from that. So I'm going to shift a little bit here and talk more specifically about the BC program and how things have been going. So a little update from our testing this season. So far, we've received almost 3,000 samples submitted for CWD testing to the program. About 83% of those have been confirmed negative, and 17% we're still waiting for results. This is a real peak time in our season where we see a real spike in hunter harvest and submissions. But yeah, so we're actively processing samples right now. This is kind of our go time. 93% of all of those samples, the total number of samples, have come from the mandatory wildlife management units in our CWD management zone. So the vast majority of samples are coming out of that area in that zone in the Kootenay region. But we are encouraging testing across the province and hope to boost those numbers. But we are getting some submissions. And then the most important stat here that I really wanted to highlight in yellow large text is that 92% of these samples have been submitted by hunters. We do, try and access samples from as many sources as we can, roadkill, collar mortalities from our regional bios. But really, hunters are the star here, submitting all, vast majority of the samples to the program, and providing all of that critical information that we need to guide our response. We're always trying to make it easier for hunters to submit samples because we rely on them so much. So, there are options for hunters if they want to retain antlers or even the whole skull of the animal that they've harvested. We always ask that the antlers are removed either, at the base of the antler or with the skull cap. But you can also submit a low jaw or learn how to collect the specific tissues that we need and just submit those for testing. Submitting those just the tissues will actually expedite result times because it saves us on our end like 3 to 4 days that it normally takes to thaw the heads and then extract the tissues. So we do really encourage hunters to learn how to collect those tissues and then submit those. We also have this an interactive map on our website with all the freezer locations. So we're adding new locations all the time. So that's a good resource to check and see where the nearest freezer is to where you're hunting. The BC lab, so all of our samples go to the BC Agriculture Lab for testing. And that lab has made some significant improvements this year to their operations and have really tightened up the result times. So we've seen this season an average of 5 to 11 days to receive a result from the time the samples are sent to the lab to when the results are posted. We've seen a bit of longer periods up, closer to that 11 days as the sample numbers have increased. You see in November is kind of our peak sample submission to the lab. But we're still, within that 2-week turnaround, which is what the lab had committed to. So that's great and much better than where we were at this time last year. A little bit more detail here on the testing process. So step one is the samples are submitted to our BC CWD program, which is run within the ministry that I work for, Ministry of Waterlands Resource Stewardship. Step two is our staff or our contractors with the program extract. We collect the specific tissues. We extract those tissues from the whatever is submitted. to us, and then we process those and prepare them for a submission to the BC Agriculture Lab for the testing. At step 3, the BC Lab runs a screening test called an ELISA, which is a type of assay that tests for the presence of those prions. And if the prions are detected, it is then classified as a non-negative, but not yet confirmed CWD positive. So those non-negative samples are then sent to a federal reference lab at the Canadian Food Inspection Agency to run 2 additional tests called a Western blot and an IHC, or immunohistochemistry. And those are the tests that are approved in Canada to confirm a diagnosis of CWD. So that is step 4. And then if that CWD infection is confirmed, the diagnosis is reported back to our BC program as a confirmed positive case. And then we will report that to the hunter if it was a hunter submitted sample first, and then we'd report that more broadly. So as many of you will be aware, last week the province announced that there is a possible case of CWD in the North Okanagan. This was from a white-tailed deer harvested east of Enderby, BC. As of right now, we are still waiting for that confirmation testing from the federal lab. So I don't actually have any news on that. We're still waiting. But at any time we receive even a possible case, we are going to take it very seriously. If confirmed, this would be a significant detection because it is so far from any previously identified cases. And it's in an area in the North Okanagan where we actually have very little surveillance data. We've had very few samples submitted from this area, so we don't have that foundational information about the health of the deer populations, or just to understand the scope of a potential outbreak here. So if CWD is detected in a new area, every situation is going to be different and is going to require or may require different considerations and different actions. But in general, the first step will be to determine if there are any additional positive cases present. Then we would determine, where those cases are to define the geographic scope of the outbreak, and then what percentage of the population might be affected. We call that disease prevalence. So in order to gather that information, we need to test animals in the area. And if hunters have provided samples and we've been able to access roadkill samples, in addition, then we can use that information to guide our next steps, to guide the response in the area. But if we don't have that information, if there hasn't been surveillance, then we may need to do targeted sampling in the area through a special hunt or a permit hunt and mandatory testing. Other mitigation measures like carcass transport restrictions would also be applied right away to prevent the potential for any infected carcasses leaving the area. The confirmed cases that we have in BC so far have all been in the Kootenay region. The sampling in 2023 and 2024 hunting seasons produced over 5,000 submissions over those 2 years. 5,000 samples were submitted to the program just in this area, resulting in a total of 6 cases. We have 6 cases confirmed in BC as of today. The cluster of cases around Cranbrook have all been in white-tailed deer. And then there was a single mule deer case further south towards the Montana border there. The cases within these 2 red circles are considered separate disease events just because of their distance and because of all of the the samples that we've tested, in between from the thousands of samples that have been submitted and come back negative, there's no indication that there's like a connection between these. So these are two separate disease events. This also, thanks to hunters in this area, the surveillance data is very strong, and we have a really good understanding of the situation. And it indicates early incursion of the disease to this area. And therefore, I believe an opportunity to contain it because we've caught it early. I want to note though that the cases in the Kootenay region, the cases that have been confirmed in BC are not the only concern, I guess, or the only potential source, because there are cases in Alberta and south of the border in Montana, Idaho, and Washington that are all very close to the border, the BC border, and could also bring CWD into BC. And so when we consider sort of potential affected areas and potential sources of disease, we're looking in BC, but we're still looking in the neighboring jurisdictions as well. So BC's CWD strategy is informed by what the experts are recommending and guided by what our partners would like to see in a made for BC plan. So it starts with that hunter-led surveillance. And if CWD cases are identified, then we know where to focus. Targeted sampling is applied in specific areas to determine that spatial distribution and how many animals are affected. Risk mitigation measures are typically implemented right away. Things like that, carcass transport restrictions, and then a longer term strategy is harvest management. And that's like a sustained effort to maintain that hunting opportunity and hunting pressure in these areas. Really important that when we do detect CWD, hunters continue to hunt in those areas. Mostly because, again, the active hunting is our most important management tool in controlling CWD. And as a secondary benefit, hunters that submit those samples provide us with really vital information to inform our response. So hunters are really key to this. So here are some of the tools that we have implemented to mitigate risk and to support the monitoring in our response. The carcass import regulation, and a number of regulations, I should say, were put in place years ago to mitigate the risk. And these are still relevant today. The carcass import regulation, the use of cervid biological materials in scents and lures, baiting and feeding. It's in Region 4 specifically is addressing that risk of animals aggregating and creating those CWD hotspots. Mandatory testing was implemented in 2019 and then expanded in 2024 after the detection, our first BC detection, along with that carcass transport restriction in the CWD management zone. We also have some best practices that are a really important part of our education. These are not regulated in most parts of BC, but we really encourage these practices to protect populations in other parts of BC. So encourage these practices everywhere. There is a general open season planned for January 2026, from January 5th to 31st in the new year and in the Cranbrook area. This will include both deer species and antlered and antlerless deer. The regional bag limit has been adjusted to 2+1. They're framing it. So that's 2 deer in the regular season plus the opportunity to harvest a third of either species in the January hunt. And the area here is quite focused around Cranbrook, so within a 5 to 10 kilometer boundary, or tightly around that community of Cranbrook. So the background is that because we have these 5 confirmed cases clustered around the city and a confirmed case within the urban deer population, um, the urban deer especially are a really high risk because, as I mentioned before, they tend to spread the disease faster and form these CWD hotspots more readily. So there is a concern that there's a source of disease within this boundary that is threatening the surrounding deer and elk populations outside the boundary. And through our surveillance data, we have pretty good confidence that the disease is not present in the surrounding populations at this time. So we want to prevent that spread. Um, there's no hunting within the city limits, obviously. So there are other actions that need to be pursued there to address that risk in the urban deer. But the goal of this general open season hunt is to limit that risk and potential spread of the disease to the surrounding populations by removing infected animals within this boundary and reducing density to help to slow transmission rates. And then a second goal of this hunt is to bolster our surveillance, particularly really, close to the city there where we don't get as many samples. It's really important that we have samples there to keep a close eye on any potential spread and movement outwards to those surrounding populations. So as we move through the response and implement these various tools, we have a continuous planning and evaluation process to make sure that we're always learning and adapting to the new information. This is grounded in science and local knowledge, as well as input from our partners. We have an amazing collaborative team. And together we will review and evaluate the information coming in, kind of zero in on the risks and make some like prioritize risks and areas to focus. And then from there, develop strategies and recommendations that we then propose to the decision makers. And then it starts again. What is the evidence showing us? What tools are available to us? Are the tools effective? And what are the communities telling us? And then we adjust as needed. So this is my last slide. I hope that tonight I've conveyed that yes, CWD is super scary and it's challenging. It's a very challenging issue, but I feel that we have knowledge and we have some good tools and we have a really great network of partners behind it. The participation from the hunting community is vital to this effort. And so far, hunters have really showed up and have been amazing. Collectively, it's going to require some hard work and some hard decisions. But I think if we're a united front, especially with hunters, I think we can keep this from getting out of control. So, I want to thank BC Wildlife Federation for providing this opportunity to share their information. And I want to thank hunters across BC for the support that you've shown this program. You have really made this work possible. So thank you for that. And yeah, look at that. I think I've made it under 40 minutes and hope there's lots of questions.
Yeah, there are lots of questions. You're way under 40 minutes, so way to go. There's a bunch of questions that keep coming up, and you normally talk about this, but it's because I asked you a bunch of additional questions. So a lot of questions around butchers, risk, cleaning tools, how effective is bleach? So can you give us just some like standard practices, recommendations, and then the effectiveness of different chemicals to try to remove prions from your knife?
Absolutely. Okay, so what public health recommends is there's sort of like two steps to cleaning. So you want to clean, and of course they recommend warm soapy water to kind of clean everything, and then you want to disinfect with bleach, and 40% bleach solution. And so that's like 40% of the bleach, like the household bleach that you buy in the grocery store. You want to cut that to 40%. And it's most effective. It's not effective on all surfaces, but it's most effective on metal surfaces. So your tools, it can be hard on tools, so you don't want to leave it for more than you have to. But 40% bleach solution for 5 minutes with your tools. If you can use metal work surfaces, that's a lot easier to disinfect than say like a wood surface, which can be really porous. Um, if all you have is a wood surface, then they recommend things like um, either disposable surfaces, butcher paper, and things that you can just dispose of after, or things like cutting boards or smaller metal trays that you can work on and then disinfect with bleach after that. So that's some of the recommendations that we've got from the research that shows that this can be effective, and the recommendations that public health has put together.
Okay, there's a bunch of questions that we're just gonna kind of maybe roll through the whole process
Hmm.
just to try to tackle all of it. Um, do we know how or why we end up with folded proteins or how that works? Or is that a longer explanation?
It's that is not my area of expertise. And my understanding is that we don't really understand there may be some people out there, I'm sure there's definitely people out there that understand that process more than I do, but my general sense from what I've read in the literature is we don't fully understand what makes these prions fold this way. Um, it's very abnormal, and the way that they fold really changes the properties of those proteins that make them um, very resistant to being denatured. Like they're just not like they will misfold and then they just become a totally different creature. Um, we know that in the body, when you have an abnormal protein, this misfolded prion, disease agent, when it comes in contact with normal proteins, they will spontaneously convert to the abnormal disease-causing prion. So I know that much. Um, we don't know where this disease came from originally, but there's so much work being done to try and better understand this. And again, it's not my area of expertise, so but this is what I understand, and what I understand from, yeah, like what happens in the body is the animal is exposed to a prion, the disease-causing prion, by ingesting those prions. And then once it's in the body, those prions will convert normal proteins to these disease-causing prions. And then that's what's accumulating in the tissue.
Awesome. There's gonna be more questions around how it's spread. And so I know a lot of it is like we don't have a handle. It's really hard to actually do the kind of tests or the kind of science that people necessarily want. But is there any science around shedding of prions in terms of how it's transmitted? Like, is there any, what's the best science around how the disease is transmitted?
Yeah, so again, we're learning more about this every day. I attended a webinar this morning that was talking about vertical transmission in utero, how infected females will actually transfer the disease to their fetuses. And so that's new information. But we know that the prion is shed. An infected animal will shed the prion through saliva, urine, feces, blood. We know that that is possible. And so through all of that bodily fluid, it can pass on to other animals either through direct contact. So grooming, nose to nose contact, mothers to their offspring through lactation can transfer the disease and infect the young. Uh, the nose-to-nose contact of males during the rut seems to be a high transmission um, period. And so there's all that direct contact, but then the animal shedding the prions just into their surroundings and it getting into the environment is a direct indirect way that other animals can be exposed. So other animals can pick up the disease causing prion from eating plant material and with prions just on the surface of plants or drinking water with prions in the water. So there's definitely that direct contact and then the indirect through the environment. And the indirect contact through the environment is one of the ways that really makes it super challenging to control this disease because then you have these kind of environmental reservoirs for disease, and so that's why um, the focus really is on staying ahead of this disease, catching it early, and controlling it before it has a chance to get into the environment. Because what's in the environment, there's not a lot that we can do to decontaminate those environments. We don't have reliable or practical tools. We're starting to get better tools, but generally we don't have access to tools to test environments for the presence of prions. So that's a real tricky one.
Awesome. Okay. What was the next thread that we were going to go on? There's a lot of questions around like, are we testing for other things, or if other jurisdictions tested for other things as it relates to CWD? Like, obviously, you have sick deer that are sick. Have any other agencies looked at what's going on with deer on the landscape that you're aware of, or are we generally focused just on CWD?
Well, whenever we have an opportunity to collect samples from an animal, whether it's a head, like a deer head that's submitted to the CWD program, or if we, have an opportunity to do a mortality investigation or something, we do make our best effort to, um, you know, honor that opportunity and learn as much as we can from those animals. And so, just with the CWD program specifically, we are collecting. Um, we are collecting genetic samples on all of the animals that are submitted for CWD testing. We have a big genomics research project right now that we're partnering with Genome BC to look at population structuring and connectivity as a proxy for CWD transmission. We're looking at genomics for CWD susceptibility. Um, we've tested animals for COVID-19. We've swabbed for COVID-19 in specific areas where we're concerned about, things like bovine tuberculosis. We'll collect additional samples to test for that disease. If an animal is submitted to the program with any clinical symptoms, then through the guidance of our wildlife veterinarian, we may be collecting other tissues, looking at things like if we have an opportunity to get organs, looking at other things like the animals from Grand Forks earlier this fall. That die off. Those animals were collected to test, for that disease specifically, but then we were able to collect CWD samples on those animals as well. So we do try and sort of make the most of those opportunities and learn the most so that we can learn more about these populations and the health of the populations that way.
Awesome. Can you, we'll switch gears a little bit and talk about the planned hunt around Cranbrook. And so I guess maybe characterizing the area, why we're focusing on that area, why it's at the time that it's at, and then any kind of supplementaries. I mean, we definitely received a lot of correspondence. People are concerned about timing. People are concerned about migratory deer. I know we've engaged the conservation officer service already a number of times to make sure that there's enforcement. We've talked to our members about this and said it's a really small area. There's not a
Mhm.
lot of areas that are crowned in this spot. It's not,
Mhm.
if you're not familiar with the area, don't go. I mean, it's basically been our advice on this. But can you just give us the background in terms of how we got to where we are and kind of how what the goal is in that area too for deer harvest?
Absolutely. Yeah. And all of those points were really good points. This is a small area. It's very close to this urban center and residential areas. Um, so it's very, very focused around where these confirmed CWD cases have been. So, of course, the once we detected CWD in this area, we were looking at, all the different tools um, that may be available to try and contain this, to try and uh, limit the limit disease transmission really is the goal. And then looking, at the broader picture of we have this cluster of cases around Cranbrook, and then based on the data that we're getting from all the hunter samples, is all of the animals in the surrounding area appear to be healthy at this point. So we need to do everything we can to protect those populations around. Um, so we really need to focus in on this cluster around Cranbrook. And so the proposal for this regulation, this general open season, was really driven by and put together by our regional wildlife team, the regional biologists that understand the area better than I do, understand these populations, where the animals are. And so they had defined that boundary and and put that proposal together, which we supported because we were working together to achieve the goals of CWD management. Um, in terms of the target for like how many animals would be harvested, I really don't know what I don't have a prediction, I guess, of how many animals might be harvested. We did last year in January, there was a permit hunt in a similar area um, where permits were issued to harvest a deer in this area. And I think we had only 37 animals harvested and submitted. So the number was actually quite low. With this general open season, I would like to see a higher number of animals harvested for a meaningful effect to achieve these goals of slowing down that disease transmission, um, removing any potential infected animals from the landscape, and reducing density. Because that reducing density in this case is we're hoping to create a bit of a buffer around that cluster of cases so that we don't have transmission and spread outside of the area. Oh, I think you're muted, Jesse. Sorry.
Sorry, I've got way too many questions going on. I know this might be a hard one because it's off the top of your head. Do you have any idea, ballpark, how many elk have been tested in the Kootenay region?
Ooh. Um, I do. I could look that up very quickly because I think sorry, I stopped sharing. I hope that's okay.
You bet.
Um,
The question someone had.
yeah, that's a great question, and I would like to answer it, and I
Okay.
will. Um, just give me 1 second here.
While you're checking that out, are you able to share with us how long we think prions might live in the soil?
Yeah, we don't know for sure how long, but there has been some work done that detected CWD prions in soil in the absence of any infected animals. After 10 years, there was still active infectious prions in soil. Um, another prion disease called scrapie in sheep and goats. They've done similar work and they've detected scrapie prions in soil after more than 15 years. So it's a very long time. We're not sure in how long, but it's a very long time. So I would like to answer that elk question, if I may. So this season, just this hunting season, we had 638 elk submitted for testing from the management zone.
Gotcha.
So
Awesome.
pretty great submission of samples from that species, which is good. It's kind of focused in on um, certain areas. I'd like to see a little bit more even distribution, just to have, to make sure that we have information from all the populations, but it's a pretty solid number. So
Awesome. Thanks. Another one, questions about home test kits for CWD.
mm,
Do they
yeah,
exist? What have they learned in other jurisdictions? How's that?
yeah, they absolutely exist. You can buy home test kits from a variety of sources, I believe. My understanding of the home test kits, that they use a kind of highly sensitive type of test that can detect very, very small amounts of prions, um, which is great. And just in general, these highly sensitive tests that are kind of being uh, have really made a huge advancements in this technology in the last few years, and I think it's going to be a really incredible tool for us. And so the same type of technology has been used to develop these home test kits. What the issue with the home test kits is that the results can still be a bit unreliable. And there's probably a range, and then there's probably better ones out there. But in working with some researchers in the States, we're very close to having these types of tools to be able to test other types of samples, like test meat for the presence of prions, um, test live animals for the presence of prions. I think we're close, but as far as I know, the reliability of those, especially the home test kits, um, is not at a level that you could have like 100% confidence in that result. And so I still have some hesitation with those types of tools. I think if hunters want to order those kits and use them and just to see, I would just caution that the reliability from what I've heard, and there's a range, but sometimes the reliability is only like 60 or 70%. And if you want to be sure of that result, then it's still best to go through the accredited labs with the approved testing, because they have a 99% accuracy.
Okay, you got the next question, because the next question was going to be, what is the risk of a false negative on initial testing?
With the approved testing, it's very low for a negative. Yeah, there's very low. The only exception there is and the reason why we don't test animals under 1 year of age in Canada is because um, the disease has to progress to a certain point for there to be enough prions in the tissue to be able to detect them reliably. And so we don't test in Canada, animals under a year of age, because in using the test that we have currently, you can get a false negative in those young animals, and so that's why that testing is not approved. Um, the Again, when an animal becomes infected, like the very, very early stages of disease, there's going to be a lower prion load, which is harder to detect. And so the diagnostic tools that we have, they're continuously improving and becoming more sensitive. And when we have access to those more highly sensitive tests, once that reliability is tightened up a little bit, I think that's really going to be a game changer for having absolute confidence. But that's why right now the testing is so stringent. And like I explained with um, our case, our possible case in the Okanagan, like it has to go through rigorous testing for us to be absolutely certain. And so there's really high standards around that.
Awesome. And then one thing that was brought up on the Cranbrook area hunt. So the question really is, why would it be in the winter? Because what's happened on the landscape? There's like the question is, why is it not in the summer? Because what happens in the winter, of course, is going to be a bunch of deer from other parts that migrate out that are showing up in that area. And so I guess the question is really, why would we focus on January, February as opposed to
Hmm.
September or something like that? And I can fill in on some of the politics on the back end too, but
Yeah.
if you can share, that'd be great.
Yeah, I mean, I think that's tricky. I think there's pros and cons to both approach. And I know that there was some really thoughtful discussions at the um uh with partners at the Kootenay hunting advisory table. And there was a fair bit of back and forth on that. But I think in general terms, if we know that animals are coming into those areas at certain periods of time, certain seasons, like, January, um, and then like if those animals have the potential to pick up disease from that environmental source or from infected animals in that area and then carry it to the surrounding populations, and we want to know, we want to sample those animals. And so that we have that information. If we don't sample the population, um, if we're missing that whole segment of that population that could be exposed when we do our sampling, then we're really missing a whole piece of the puzzle.
Awesome. Okay, is there, and you normally touched on this again, and we kind of, because we changed it, is there a vector other than ungulates for moving prions across the landscape?
Yeah, so and I should clarify, it's just not all ungulates, but cervids
Cervix.
or species in the deer family that are known to be affected by CWD. There's no indication that it can infect, sheep or goats at this point or other ungulates. Um, but yeah, so the animals, live animals or infected carcasses are a vector, but it can also be present in um, you know, other environmental materials. Like it can be present in soil, it can be present on plant material, in hay, like hay. It's theoretical because we don't have a way to test hay at the border, but we do know that in some areas where CWD is prevalent, the environments are contaminated, and then plants are harvested and transported to all kinds of places. Um, so that movement of hay, even you know, soil, dirt on equipment or trucks in the back of trucks, um, mud on your boots. I mean, it's all potential. It's all could potentially be moving prions around.
Gotcha. And so the next question talks about that, and if you don't have a good answer, that's great. Question is really around, why did the cluster start right near Cranbrook, and could it be because a hunter brought a carcass in from Alberta or from Saskatchewan, I know it's going to come up if we do end up with a positive in the Okanagan as well. And so if you want to
Mhm.
use your knowledge or if you want to take a pass, those both work.
No, I'm happy to answer that. The bottom line is we don't know how CWD entered the Cranbrook area. We may never know. But there, we are looking at, the different potential vectors that could have brought it there. And absolutely an infected carcass is a possibility. Um, or contaminated hay is a possibility. And based on what we're seeing in the Cranbrook area, it could have been. I mean, we're within natural animal range that an animal could have walked up from Montana and set up camp, or just hung out with some deer in Cranbrook, and then went back to Montana, and that's how it started. But you would expect to see more evidence of cases sort of between the two disease, like affected areas, where we're not seeing that. We're kind of seeing this cluster, which, again, I don't know for sure, but it does sort of suggest to me that there, could be a point source that was brought into the area.
Awesome. Any update on the genomic work?
Yeah, the genomic work is ongoing. We collected over 1,000 samples from mule deer and white-tailed deer last season. And that work, so those tissues were sent to a lab in Quebec where they were genotyped, and now we have a postdoc working on that now, analyzing that data and looking at the landscape genomics, looking at relatedness, looking at potential population structuring. And so the results are still preliminary, but that project is really progressing really well, and we hope to have more to report in the next like 6 months or so.
Awesome. Okay. We got a lot of, we're at just about a half hour worth of questions. What else do we have? Prepare deer out. I think most of these questions you covered during your presentation. Um, Salts, all this stuff has been. Yeah, and so this is a good one. Again, we're kind of going back, but what if jurisdictions that have had COED for decades found to be effective methods? So if you have it at the top of your head, if you could just go like Alberta, Saskatchewan, Manitoba,
Mhm.
Colorado, Montana, and just talk about what they've experienced because it's highly variable depending on the species. And maybe just to lead
Mhm.
into that, are there cervids that seem to be immune to it, and are there populations or sorry subspecies of cervids that seem to be more impacted by CWD than others? Let's start with that, and then just work your way across the prairies and then down through the states in terms of prevalence, population stuff, if you don't mind.
Sure. Okay, so the question about are certain animals or species, less susceptible. Um, we don't the research so far isn't indicating that there's you know genotypes out there or certain populations that are less susceptible, but there are um, there is sort of variability in how animals are affected by CWD, and there's different CWD strains as well that contribute to, sorry, sort of like variable outcomes. All the animals will always die, but sometimes it takes, they will die over a shorter period of time. That type of variation in sort of the disease progression. Um, in terms of different cervids affected, there is some really interesting work that has demonstrated that certainly in deer, we've seen that once the disease reaches like a 40 or a 50% prevalence, that's when you start to see dramatic population decline. But what they are seeing in some elk populations is you start to see that decline earlier. So so prevalence in elk of like 25, 30% is sort of the threshold for when you start to see the population level impact and number of animals declining. So that's concerning, for sure, to see that kind of increased impact or potentially an earlier impact in elk, for example. Um, so I like moving across, it'd be kind of hard to go like jurisdiction by jurisdiction, but I think broad strokes is, in Saskatchewan and Alberta, unfortunately, they were hit with this disease really early when we didn't really understand the disease. At that time, we didn't know that it would contaminate the environment because other prion diseases don't do that. And so just how that management was approached, they just didn't have all the information. Um, and, to their credit, they really, tried to control this disease, but maybe made some missteps along the way that unfortunately led to losing support for their actions and kind of derailed their management effort. And so there wasn't support for management for many, many years. And during that time, the disease established and kind of got to that point where management would no longer be effective. Um, and so as we move through, and that was kind of the case in Wisconsin. The Wisconsin-Illinois comparison is really interesting, because they detected their first cases around the same time and had very different approaches to the management, where Wisconsin went in quite aggressive, similar to Alberta, and they, just kind of lost support from hunters and from the public, and they lost that political support. So the management programs were discontinued. Whereas Illinois, I think, really took a bit more of a much more of a thoughtful approach and sat down with hunters and partners and mapped it out. Because if you don't have that um, collaboration and cooperation that you're going to need to sustain for like many, many years. The Illinois program has been in place for, over 20 years now. And so you really need everyone to be on board and supportive of it to sustain it for that period. So that was really key. But certainly what we're learning now too is that detecting the disease as early as possible is absolutely key. I mean, once you get into even, over 5% prevalence, your management, the effectiveness of management is just going to start to decline. It's going to get away from you. So those proactive efforts that Manitoba applied with mandatory testing. Manitoba and BC, we were kind of, we were watching what was going on in Alberta and Saskatchewan, and we really made an effort to be proactive to build those surveillance programs so we could catch it early. And similar examples in the States where that has been done, that really seems to be key to this. And I mean, we're still early days, Manitoba is still early days, but some of the evidence from other places that are a little bit further along really indicate that is key to get on it soon. So yeah.
Yeah, well, thanks, because it is, we work federally and run into all of our colleagues across the country when we're in Ottawa, and we have these chats all the time. And of course, like Alberta is really bad, and everybody points to it and says it's really bad. And yes, it is. And Saskatchewan is really bad, to the point where the people who come from southeast Saskatchewan said they don't apply and they don't hunt near their houses anymore because the prevalence is so high. But Manitoba border, which is where Manitoba had, I think, 2 positive cases. They did a small 10-kilometer square, I think, targeted reduction, and their prevalence is like 0.1%, right? So they're
Yeah.
4 or 5 years later. Whereas Southeast Saskatchewan for mule deer, like the the ED was telling me they're like 90% or 80 something percent prevalence for mule deer bucks now.
Yeah.
So it just tends to get away with you. So
Mhm.
anyways, Cait, thank you. We've taken up more time than we would have expected. Thanks everyone for all the questions, tons of engagement. We do have, webinars from before too. We have webinars from folks in the United States, Wyoming. They did a webinar for us, and it was the same thing. They looked at harvest rates and prevalence rates, and what the paper tends to suggest is that if you have moderate harvest or high harvest, especially on bucks, that you can keep prevalence low, and if you don't do anything, you end up really high really quick, and so there's no fun. Maybe one thing, if I'm not sure how up to speed there is work around trying to find solutions, but it's not working. The solutions are not working yet, but there are people that are trying to work on solutions. And do you want to touch on that before we close out, or just to give people a little bit of hope so that there's a there's attempts to try to deal with this?
Well, yeah. I mean, I think that that's the message here is there's some huge effort, a huge body of work that we can learn from, and a huge active effort to try and find solutions and to approach this in a thoughtful and measured way. And I think there's lots of opportunity. I think BC is very lucky. And there's some other jurisdictions that, are in the same position as us right now. But BC is very, very lucky with our very low prevalence so far. And so I really do think it's an opportunity to apply everything that we've learned. There's experts from around North America that have reached out to me and have said, BC is going to be a great test case because you've caught this early. We've got good tools. I think you have a chance here, and so I'm hopeful. But we got to work together. It's going to be hard work, but I think we have a good opportunity.
Yeah, awesome. Well, thanks, Cait. Really appreciate your time. Thanks everyone for tuning in. I don't know, there's probably closer to 200 people between here and social media. So awesome work. Thanks to all the work that you do and your team does. It's phenomenal. We are constantly advocating for more resources and trying to make the system move quickly and be responsive. So we appreciate all the hard work that you do put in. And yeah, we'll talk again, I guess, probably this year in the fall and get an update and see where we're at. Philippe did post a link to a video in the chat as well, which gives people a way to prepare for their harvest for a CWD test and a Euro mount. So now you've got that tool for folks in the Okanagan and the Thompson. We're waiting test results on that one, but all it really does is tell us that there's a whole bunch of deep freezes that have been voluntarily put across the region and all over the province we're working on. And also iHunter was mentioned here. iHunter's done a great job, and I see Jeff Gailey's on here. He reached out to them and they put the depots, like the freezer drop-off locations, are actually on iHunter. So if you're ever out hunting and you want to find a depot and drop your head off, that's a place. And then somebody was also saying the Cranbrook management zone is also on iHunter. So you want to check that out, please do it. As said, it's a small area, a lot of private property, a lot of municipal land. If you're not familiar with the area, it's not great, and we have definitely reached out to the regional staff and the conservation officer service. So anybody who's considering going should expect a heavy presence from the conservation officer service to make sure that what we're trying to do in that area is being conducted in that area and being done properly. So thanks everyone. Appreciate your time. Thanks, Cait. We'll get you a green
Thank
jacket for all
you.
of your attendance, and keep up the great work. Thanks, Philippe, and everybody
Great.
at BCWF. And I hope everyone has a good night.
Great. Thank you, everybody. Thanks, Jesse.